In today's class, we will study preclinical research methods in drug development. This topic is present in BAMS third phase syllabus in that research subject and in the fifth chapter, eighth topic. Now here the topic is preclinical research. What is meant by preclinical research? It means that before giving to the human being, the new trial drug is tested on the animals or the other in silico in vitro. That is called as a preclinical methods. Here the majorly four methods we are using to test in the form of in silico method, in vitro methods, in situ as well as in vivo methods before giving to the clinical or human being. Now we'll study what is meant by preclinical. Preclinical research is the initial phase in the drug development conducted before trial. It means that the before giving to the human beings, this is one of the initial phase of the drug development, the new trial drug. Here, what are the things here we are evaluating? First is the safety of this, whether the drug is safe or not, efficacy, whether the drug is effective or not, pharmacokinetics, it means the ADME we are studying here, toxicity, whether drug is toxic, of new drug, the candidate that we are testing here in the methods of such as the in silico methods, in vitro, in situ, in vivo studies we are conducting in this phase. Now we'll study one by one what is meant by in silico method. The first term is in silico. In silico is nothing but this is the computer chip is called as a silico. In silico is nothing but the inside the computer. It means without using any humans or animals, that only by using the computer or only the using the computer chip, we are predicting the drug, how it is effective, how the drug is working, that we will study in this phase. In silico means the analyzing the computer based studies. It means that is analyzing the predicting how the drug will affect. The principle is use of computational stimulation to predict drug is target interaction, ADME properties and toxicity. Before other methods using this drug is firstly we are using for predicting the target interaction, the what is actual where you want to target, the whether the drug is targeting or not, that we are analyzing the interactions, the combinations as well as ADME, the absorption, distribution, metabolism and excretion property also we are checking through the computer, as well as the toxicity also we are analyzing using the in silico method. Okay, and now we can take very good example here, the molecular docking of curcumin. You know that is the curcumin is one of the extract that is curcuma longa. In simply it is called haldi or haridra in local language, haldi. In that the phytoconstant is present chemical, that is curcumin. Now how we are using this computational technique, that is in silico methods, you know that is whenever there is a body in that inflammation it is there, the COX-2 enzymes is the due to the COX-2 enzymes the inflammation you can see. So whether the if you give the this curcumin, whether it is effect on the COX enzyme or not, that we are predicting by giving the analyzing in the computer. Just we are entering the curcumin here. Docking is nothing but the combining. The molecule of curcumin is combining with the this in the computer based studies. Here the COX with COX enzymes, you know that is the COX-2 enzyme is a one of the inflammatory enzymes. Whenever the inflammation is there in the body, this enzyme is the responsible enzyme to predict the inflammatory activity, whether the curcumin is the helpful for the acting as an anti-inflammatory drug or not, that we are analyzing using this computer technique. This is the one of the very good example in Ayurveda. Okay, now the next one is application where we are using this in silico method. The mainly the molecular docking. Just now I told the molecule is nothing but the new trial drug here. The docking is nothing but the whether the drug is the uh, combining or interacting with the our target that we are analyzing. It means that whether it is correctly working or that is a combining it not, that is we are checking, that is the docking. Next is the visual screening. What is meant by visual screening? The lots of the molecules we are passing through this in this computer, which drug is that is combined with the particular target, that drug we are using for the next stage or for the animal trial or human trial. It nothing but the visual screening is nothing but the so many thousands or lakhs of the drugs we are searching in that, which drug is in the computer shows the effective, that we are separating and using. This is the visual screening. Next one is the QSAR modeling. What is meant by QSAR modeling? It means that quantitative structure activity relationship. It means that the similar drugs we are using, very good example that omeprazole is one of the PPI drug. The similar structure the scientists use to produce the pantoprazole. Okay, like that, from the pantoprazole again rabeprazole, esomeprazole, like that the similar structures the scientists using for new drug development, for the development of new drugs. This QSAR modeling technique, we are using that similar structure drug, something only little bit will change whether drug is effective or not. We are analyzing by using in silico method. This is also one of the best application through the in silico method. Next is the advantages. Why the in silico method we are using in the research? The mainly it is a rapid, fast acting, ethical. There is no need for permission for any from anywhere, animal committee, ethical committee, or human ethical committee, and cost effective. That is does not require much money to for spending. This is a very cheap. Limitation is the based on the models, because this is only we are analyzing based on the models only. That's we are not at all using the any living organism. That's why this is one of the limitation. It is there may lack of full biological accuracy. It may be not accurate like the any other trial. That's why these are the limitation in the in silico method. This is about the in silico method.